Good afternoon all, and welcome to the Scientist.com webinar, Overcoming Challenges in UK Biobanking. Part one of our series is focused on maximising the use, value and impact of human samples. This webinar is presented in collaboration with the UKCRC Tissue Directory and Coordination Centre, aka UKCRC TDCC. Today, I'm going to give a brief introduction followed by a presentation on the challenges in UK biobanking and ways to help biobanks increase access to their samples in a transparent and compliant manner. Our fantastic panel consists of Richard Stevens, patient advocate at Use My Data, Genomics England, NCRI and a few others. Helen Hind, clinical samples manager at Medicines Discovery Catapult, and Dr. Phil Quinlan, director at the UKCRC TDCC. After the presentations, we will have a question and answer section to give you an opportunity to engage directly with the panel. So please feel free to type your questions into the Q&A box at the bottom of your screen as you think of them, and we'll be sure to answer as many of them as possible. My name's Matt McLoughlin. I'm VP of Compliance and Categories at Scientist.com. I've been involved in drug research and discovery for the past 20 years and think every day has involved human biological samples in some capacity. Within my role at Scientist, I'm responsible for the creation of our award-winning Comply platform that supports most of the world's largest pharmaceutical companies access human samples from commercial and academic biobanks. I also lead a team of experts who support research on a daily basis to identify and source the resources they need to drive their experiments forward. To give a bit of background as to who Scientist.com are, we're a software solution to support scientific research that was founded in 2007. We're headquartered in San Diego area. However, as you can probably tell, I'm based in a much more desirable city of Manchester, and we have offices all over the world, including the UK and Japan. We currently have approximately 60 to 70 full-time employees supporting over 80 biopharmaceutical companies with over 3,300 suppliers in more than 4,000 different research areas. The aim of Scientist.com is to facilitate direct interactions between industry researchers and providers of samples and services in an efficient and effective manner. I should be clear that we are not a commercial provider of samples. Our platform enables research organisations and biobanks to ensure all samples are requested, offered, provided and received according to your own internal policies and procedures. It is for this reason we have supported thousands of requests for samples from researchers across the globe to from a range of commercial and academic sources. I'm very pleased to be able to introduce our fantastic panel today. Our first speaker is Richard Stevens. Richard has survived 2 cancers, a heart emergency, and continued comorbidities and late effects. He has participated in 4 interventional studies and 9 others, and has donated tissue and samples for research. Richard chaired the NCRI Consumer Forum and now chairs BBMRI ERIC's Stakeholder Forum. He sits on strategic groups for NIHR, NHS England, PHE NCRAS, Genomics England, Cancer Research UK, and the ABPI, and works with industry and with patient advocates globally. He is the patient lead for the UK's Living with and Beyond Cancer research priorities, and co-editor of the journal Research Involvement and Engagement. After Richard will be Helen Hind. Helen has over 25 years experience in life sciences, having previously held both lab and clinical research roles at AstraZeneca. She has also worked within sample access for AstraZeneca's discovery scientists. Prior to her current role, Helen worked in academia on clinical trials methodology and within the NHS as a business development manager, supporting commercial research in the Liverpool City region. She leads Medicines Discovery Catapult's samples and data capability to help UK drug discovery companies, primarily SMEs, access the clinical samples they need to develop medicines faster. And finally, Phil Quinlan. Phil's main responsibilities are to oversee the strategic direction of the UKCRC TDCC and to ensure delivery on its mission and objectives. The team works to maximise the use, value and impact of the UK's human sample resources, helping ensure that UK biobanks are visible to the research community. The field is quite complex with a wide range of stakeholders, and part of this role is to ensure that we understand the various needs and concerns of the communities, as well as the potential opportunities. His background is in application development and data analytics with a PhD in AI and health informatics. His expertise is around data systems and data integration to assist biobanks in connecting the wider world. With those introductions complete, welcome to the webinar. And now I'm going to hand over to Richard to the first presentation.
Thanks very much, Matt. Good afternoon, everyone. Let's have my first slide, please. This sets out some headlines about what I want to discuss for the next 5 minutes. And I must emphasize that although I've been described as sitting on lots of bodies and a member of lots of groups, my prime interest in this is simply as a patient who wants to benefit research, as a patient who has benefited from research, and as someone who has donated tissue and samples and was quite shocked when I discovered that some of the samples I donated 20 years ago are still in a collection somewhere that haven't been used, and more to the point, the collection has never been advertised to anybody else, so no one's likely to use it. And that's why I got involved with this particular project. So there was a workshop last autumn containing lots of stakeholders in the biobanking world, and it produced a report in January 2020 called The Issue with Tissue. And the first bullet points are from the report, and the web link on the slide is where you can access the full report. What came out of this was 2 points of principle and 10 action points. The 2 points of principle really go back to my own personal shock, which was shared by other people. Every tissue sample that we collect in the United Kingdom, or anywhere else for that matter, should be discoverable. People should not have private collections. And the success of biobanks should partly be assessed by their usage rates. And I know there are collections on clinical trials, for example, which are stored for donkey's years because what we're looking at is, or what we're preparing is for people with research questions in 10 or 15 years as part of a pipeline of research. That's fair enough. But nevertheless, we should be looking at usage rates in biobanks. Are the collections being used for what they said they were being collected for? Here are the first 5 action points. And while I'm talking, I hope people get the chance to read through them. And with luck, people will read the first couple maybe and think, well, this is correct. This should be happening. Why isn't it? And then we get down to point 4, which is one of my own personal ambitions. One of the ways of making this happen is to have one unified national directory and make it mandatory to register the collection. And certainly funders should be asking where the collection is going to be registered, and ethics committees should not be approving collections unless there is actually some indication that people will register and thus advertise what the collection is to people likely to use it. And I'm delighted to say that Cancer Research UK has decided it is not going to fund tissue collections unless there is some sort of dissemination plan that is visible to the funding committee. Their funding committees include patient representatives. So the last point there, common ways of cataloguing and publicising samples. As a patient, as a lay person, I sit back and think, why on earth aren't they doing this already? I'll take another pill to control my blood pressure. And here we are, the second 5 points. Publishing the turnover rate, which might be high, it might be low, it depends on what the bank is set up to do. But that's why you need to publish the figures and a commentary. And we all need to be aware that different rates are appropriate for different banks. The point is, the banks are now in public. Everyone can see, including the people who've donated their tissue, what's going on. Standardization, transparency, and then we get to point 9, and this is something that industry could join in with. How about acknowledging our contributions in any research publications? It's our tissue, they're our samples. We gave them to you, the biobanking community, to benefit humanity. So let's just have some thank yous published, shall we? And that in itself will help point 10, because as we've seen with things like clinical trials, patient-to-patient conversations is just one good way of widely publicising all of this. And there we are. 2 quotes from colleagues of mine, fellow patients. One is really simple, that if we give our tissue or samples, we want something to happen with it. And if they are being deliberately held for a few years until a particular line of research comes up, then fine. Just say so and make sure we understand that when we're giving our consent. And then the second one is a person who said they don't want to give consent, they want to give an instruction. When I give you my tissue, I want it to benefit others, so use it. Thanks very much. At this point, I'm going to hand over to Helen.
Thanks, Richard. So I'm just going to talk briefly through some of the factors that I see as being relevant to SMEs around sample access. And by SMEs, I'm referring to the small and medium-sized UK organisations that are working in the life sciences sector. But just for a bit of context, the reason why Medicines Discovery Catapult has a sample access theme is because several years ago, when the organisation was in its very much infancy and setting up stage, we were identifying key areas of focus to support the life sciences industry in the UK, and produced a State of the Discovery Nation report in 2018. And this surveyed UK SMEs around the various challenges that they had within R&D. And one of the messages that came back quite strongly was that access to high quality clinical samples was something that was extremely important to them, but in reality it was difficult to achieve. So then, thinking why SMEs need samples, they need them as part of their core R&D activity to progress their assets in development in order to bring new drugs and diagnostic products to market, which ultimately will deliver patient benefit. And they can get these from a number of sources. They might obtain samples through setting up collaborations with clinical academics or other key opinion leaders where they have those connections. They could look through NHS and academic biobanks and use the UKCRC tissue directory to identify some of these. Could work with commercial sample supplier organisations, for which there are a number based in the UK. And they could look at working with other platforms such as Scientist Marketplace. And typically just some of the things, some of the projects I've been involved with, what they're using those samples for, it might be for validation work, so to validate an assay that they're developing, for other method development work, in sequencing assays or biomarker analysis. They're often interested in comparing normal and diseased tissue, often from the same patient, or collecting matched samples, for example, blood or PBMCs and the matched tumour tissue. And from the information we've gathered in MDC over the last 2 years, over half the requests we've seen have been for oncology samples, but really all major disease areas are represented: neurology, infection, inflammatory disease, respiratory, etc. So right across the spectrum. So then thinking about some of the barriers to sample access in the UK, and this is from surveys that we've done in recent years and also from individual feedback that we've received and collated. The UK has rich sample resources that are stored in numerous biobanks, but many of these are never accessed, as Richard had already alluded to, and it's trying to understand some of the reasons by that. In some cases, SMEs may not know about them, particularly if they're a very small early stage company and they have limited experience in sample acquisition. This is different to large pharma organisations who often have large teams of people who are experts in this area that work in sample acquisition. Some of the biobanks aren't adequately promoted and they can be hard to find if there's no central website or if they're not indeed listed on the UKCRC tissue directory. There is a national infrastructure to some extent through UK Biobank and Genomics England, but these have tended to prioritise large pharma organisations and are not really focused on the needs of SMEs specifically. And because there's no standardised structure, the issues encountered can be with the procurement itself, through negotiating often complex requirements of access committees, the speed of access to the samples, or other governance issues such as ethical approvals. And then moving on to think about how biobanks might overcome the barriers. When I've spoken to a number of the biobanks that we're looking to work with, the main aspect that the biobanks will cite to us is it's all about having a robust cost recovery model, which is important. Although every institution will approach cost recovery differently. You can list examples of different items that can be included within that. But we feel that there are other areas that do need consideration. So things like customer awareness, how do customers actually find their biobank? Are they listed on the tissue directory? Are they listed on other platforms? Do they have their own website, or is that embedded within a trust or a university website? It could be quite hard to find. Around customer management, industry like a prompt answer, and particularly for SMEs, they have to move really quickly to get their products to the next development stage, to reach that next investment decision. They like a prompt answer, even if that answer is no, so they know to look elsewhere. They need to know quickly if the request is feasible for the biobank or not, and get an early read on that before they're completing an access request form. Also important to consider the operational processes. So how do you apply for sample access? Is there a clear process on the website? Are the request forms available to download? Do we know anything about the composition of the access committee, how they meet? Is it a virtual meeting at set intervals? Is it via email? And just having some clear expectations on timelines. What's the level of patient and public involvement representation on that access committee? Then thinking around the documentation, so material transfer agreements, etc. Are they publicly available? Can you see what the agreements look like? Are the terms negotiable in any way? For example, some of them may have intellectual property clauses that are too restrictive for SMEs to be able to sign. Do they have delivery resources there? So are there dedicated people to manage the end-to-end process? So is there a business or biobank manager? Are there sufficient technical staff to process requests, and who is responsible for managing that? Who's got the authority to progress those requests? And then the donor engagement, and again, as Richard's mentioned, patients do want to know that the samples that they've donated for research purposes are in fact being used, and some feedback at the end of the project on that and acknowledgement is extremely useful and does maintain patient engagement over the longer term. So I'm thinking about who could act on this and get involved. In terms of the sort of funder level, I would cite, two, the Health Research Authority and the Human Tissue Authority. So for the HRA, they have an agenda around transparency in research, yet despite the registration on the UKCRC Tissue Directory being a condition of approval for a research tissue bank, we know in practice that it doesn't always happen, and that resource isn't findable. HRA say to us that they don't see themselves as being the enforcers within this process. They would more likely get involved, for example, if there was a serious breach of patient consent. Similarly, the Human Tissue Authority, they're involved in various areas as well as research, such as transplantation, anatomy and post-mortem. And research actually makes up a relatively small area of what they oversee. And while they've said to us they'd love to see a high proportion of samples held in UK biobanks being used in research, again, they don't feel it's their place to intervene. They'll be more likely to get involved if organisations were in breach of the Human Tissue Act in respect of sample storage or the operational processes that they were undertaking. And patient groups are, understandably concerned and disappointed about these low use rates. And patient groups increasingly do need to make noise about this, about what the issues are around use rates, and they need to be listened to. And then finally, just the importance of utilising UK bioresources. When biobanks are set up, many of these are set up initially with grant funding, and that may be for a short number of years, but ultimately they will have to demonstrate sustainability and will have to attempt to do full cost recovery. And if that doesn't happen, biobanks could potentially end up closing down, and then you've got a further issue in repatriating those valuable sample collections elsewhere. And the other aspect of that, I would say, is the keeping that process within the UK delivers a benefit to UK PLC. So yeah, it's something that, in summary, it's something that SMEs need, and it's something that we need to see happen at scale within the UK. It's hard to understand why we haven't moved on more than we have, but I'll probably leave it for there. And if I could hand over to Phil Quinlan.
Thanks very much, Helen, and good afternoon, everybody. And it's great to see so many people joining and engaging in this topic, which I think is really important for us to do. And I'm really pleased that we're having this conversation in the open as well, to show that we're keen to get a solution out there and to have transparency around this. So I thought it'd be useful just to set out where the UKCRC Tissue Directory and Coordination Centre came from. And it started with a very positive statement from a group of funders. So if you're not aware of the UKCRC, it's the UK Clinical Research Collaboration, and it's where funders can come together to discuss topics that they think are challenging for all of them. And they created this funders vision. This was back in 2011. And this was really the foundation point from which we were then funded to try and tackle some of the challenges. And in particular, the one that's on the screen there was the one that we were set up to do. And the key parts out of that were around minimizing duplication. So there was a feeling that there were many biobanks out there that were collecting samples, but the funders didn't necessarily know how many of them were there and whether samples already existed in biobanks that could be used by researchers now applying for funding to collect more samples. So they realized they needed a system to make sure that they were only funding the collection once and those samples were used as widely as possible. And one of the main ways they wanted us to do that was to create a tissue directory. And that's where that phrase easily discoverable and accessible comes from. So it's how do we make people aware of what is available? Because obviously we can only reduce duplication if we actually understand what is actually there. So as the UK's national centre, we now have just over 200 sample resources registered in the directory. We've plotted the cities that these are present in, so you can see that we have a very good coverage across the UK, and we have a good representation of people there. Some of the points that have been brought out already by the other speakers. So we are now a condition on registering. So if you apply to be a research tissue bank to your ethics committee, it'll be a condition on there. There are some improvements we can make there, as already highlighted, around how we reinforce that message and get that through. But we now do have quite a number of biobanks on our directory, so it's a good basis that we've got there. The really interesting fact is nobody knows how many are out there. So we have got just over 200, but we don't know where the end game is because there's no single entity will actually know the answer to that question, which is obviously very interesting. The directory itself is not necessarily just purely a sample catalogue, so we don't just catalogue the samples. It is around the data sets and the population that's available that could be supporting research, and as well as the team that's based at the University of Nottingham, which do the development of the systems. We have a team at the University College London who do a significant amount of user engagement work to understand what can the biobanks realistically provide, what can they actually do for us, but also what do researchers need, and that's very much a balancing act between those two communities. So it's trying to understand what's possible and what's needed, and balancing between those two areas. And really, we see this as a continuous piece of work. It's not as if we can just create a directory, job done, off we go. Research demands change, situations change. I think the last few weeks or month has definitely taught us that, and we're trying to respond to COVID in the same way. And so we constantly need to be evaluating and having discussions. This is why we're very keen to support webinars such as this, is to understand what's needed and how we can help. We are also the nominated node within BBMR-ERIC, which is the EU infrastructure for biobanking. And I think it is important to acknowledge, whilst we are talking about the challenges in the UK and how we can address those, these challenges aren't a unique UK problem. So we do see these across multiple countries and worldwide around how do we get samples used. So the low utilization rates that may be quoted for the UK are applicable everywhere. And it's really how do we work together across the communities to try and change the dynamic and make sure biobanks are there and used and well represented. And I think one of the key issues for us is around misuse, in the sense of you've heard how governance and regulatory bodies will respond to misuse in the terms of something was done that was outside of the consent, so a serious breach of consent, or something wasn't in line with the HTA. But actually, probably the largest misuse of samples currently is the non-use. So if you think about the consent that was taken, and you think about that, particularly that phrase really hits me around, it's not consent, it's an instruction. How many of those instructions are followed and how many are not? And that's not just to be critical of biobanks, because actually one of the things that we see is that the lack of data is one of the biggest reasons why samples aren't used. And if you try and, at the moment, try and link clinical data, you have you'll have a whole range of problems and challenges that you'll experience when trying to link to clinical data. So where we've got a patient that's made a very generous donation to say, use my sample to improve healthcare, that sample is collected, but then we can't link the data in order to allow that sample to be used. Is that a breach? Is that a misuse? And I think we need to really think about where we set that risk threshold in some of these conversations. So picking out some of the work that's been undertaken by colleagues at University College London, so this was from Emma Lawrence, we tried to understand where were the major blocks in the system. And I think it's where you've heard from Helen in relation to SMEs. This was more of a survey from academics replying. So when we asked them what are the barriers for accessing human samples, you can see governance comes out quite strong. So ethical approval, MTAs, contracts as quite significant barriers. But also the fact of just locating them, knowing where they are, is also a barrier there. And as I hinted earlier, if you then think about why are samples not actually used in research, the largest barrier there is the lack of linked clinical data. So we really have to think about how do we get this whole system working. So it's not simply that biobanks don't necessarily want to give samples out, but they don't necessarily have the samples and the data sets that researchers require. So how do we change that dynamic, and how do we make sure samples and data sets are used for the research that they were donated for? And one of the things that we have tried to do is to reset the thinking around how industry might engage with particularly academic biobanks. So we released some transparency principles, which was really to get companies who can help those connections between SMEs or pharma and biobanks to agree how the relationship should work. And really what we've been concerned about before was organizations that would maybe blind that process, in the sense of they might have an agreement with the biobank, they would have an agreement with the end user, and the end user and the biobank would never know who each other are. So obviously, if you're trying to report on use, track samples, give any sort of reporting back to patients about what the samples have been used for, that's very hard. So we set these transparency principles out particularly to challenge this. I'm really pleased that scientist.com were one of the first organizations to sign up to these principles, so thank you very much for that. That was really good from our perspective. And also we thought it was important, so we added the use my data component there to actually always acknowledge the patient donations that were made in any publications. So there's principles there, both for the biobank, the organization that's enabling the access, but also on the end users to make sure that the transparency is all the way through. And that's it from me. So thank you very much, and I'll hand back to Matt.
Thank you, Phil, and thank you all for the very informative presentations. I hope the attendees found those brilliant. Before we go into some of the questions, I just want to take this opportunity to remind the attendees that if you would like to submit a question, there is a Q&A box at the bottom of your Ring Central Meetings control. You're able to submit there. That will come to the team within scientist.com, and then it will pass through to me, and I'll be able to field it to the team. So if you do have any questions from anything that you've heard from the brilliant presenters today, then please submit. We have, however, had a couple through already, so we're going to kick off and go straight into those. So first of all, the first question is, what is a good utilization rate, or what makes for a good biobank? Is being empty realistic? So not to put anybody on the spot, but Richard, do you want to take a first pass at that?
Thanks very much. I wouldn't necessarily make that judgment. I think the question is slightly wrong. I don't think is being empty realistic. I think is being empty, is it desirable? If you have a tissue bank of tissue that's been collected for a specific purpose, is it actually being used for that purpose or not? If you have a more general bank, that's a different issue. If you are a prospective collection, and I know there are one or two in this country now going out to collect samples on request, clearly you're going to start off empty, you're going to fill up, and then you're going to be emptied again. So I think for me, this is why the issue for tissue says that we don't just want the figures published, we want a commentary on why your figures are whatever your figures are. The big challenge is not whether or not a tissue bank is empty, it's whether or not it's still got a whole pile of samples that nobody knows about and or that nobody's touched for 10 years, precisely because perhaps the data isn't available. Yeah, why are we still sitting on that? Why do we collect stuff and not tell people about it? Those for me are the big issues. After that, we start talking about usage more generally.
Helen, Phil, anything to add? If not, then we can move on to another question. In which case, this has been submitted. What could industry do to help biobanks make samples available to them?
Yeah, so we see this on a reasonable regular basis with the directory where biobanks are on there, obviously, and industry come along and approach them. And I think there we need more discussion, more open discussion around the needs of industry, and also around how you actually would engage with biobanks, because sometimes the expectations from industry can be too much sometimes on biobanks, in particular what their requirements may be, their timeframes, and understanding how a biobank may actually be funded and set up. So we did mention earlier that a lot of biobanks start as an academic project and then are converted almost into a service, and that can be quite a hard process. So I think it's around having that dialogue, trying to understand what the demands and needs of industry are, and how that can actually support the development of the biobank industry type thing in the UK. So I think conversations, transparency are key to that.
Yeah, I think just to add to that as well, we within the scientist.com group, what we noticed that there was certainly a lot of hurdles that industry wanted Biobank to jump through. You know, they wanted to use their own contracts, they wanted to utilize their own approval process, so they do quite a lot of due diligence on those biobanks, which can actually be quite prohibitive. When we looked at this back in 2016, 2017, we noticed a lot of duplication. So when a biobank was working with an industry partner, they were having to do the same work over and over again individually each time. So one of the things we've been doing is looking to try and remove that duplication, and we're very proud that we've now supported, as I mentioned in the introduction, thousands of requests. And one thing I didn't mention is that all of those requests have been done under the exact same contract. So all of our industry partners, all of the providers of the sample actually just work under one set of terms and conditions, which addresses the requirements such as the material transfer agreement, addresses the ethical considerations and things like that. And I think to really open up the biobanks and the samples and get those samples to the researchers, we need to be doing more ways of standardization, because I think it's the variability aspect that is involved, which can lead to a lot of delays, which was also sort of shown through Emma's data earlier. One of the questions we've had, then I'll leave this one open to the panelists. In cancer, using a clinical sample without understanding the clinical background with the help of the clinical data is quite dangerous, as it can potentially lead to false conclusions. Why is there not more being done to facilitate the linkage between samples and clinical data?
So I can try to answer that from a patient perspective, a cancer patient who has donated samples. I think trials that are being run now, the linkage is made in the trial structure. The problem is often that the patient is having their trial as part of their treatment. It's a treatment option. We have an issue which has been raised in this discussion, that consent for sample donation isn't necessarily explicit about future medical data that might be linked to it as you track the patient through their pathway. There are patient groups that are working on this. There are patient groups helping the HRA, NCRI, and other people looking at generic forms of consent. There are patient groups helping NHSX make it easier to release clinical data. So there is an awful lot of work going on. The problem we now have in the UK is there is a trend that we don't like centralization, but against that we now have loads of different organizations who are all charged with looking after bits of data about me, the patient, and they're all trying to act in my best interest. They're all trying to preserve my privacy, and the result is that it is now 3 or 4 times more difficult to get hold of data and to link data than it was, say, 10 years ago, even though ironically we've got a heck of a lot more data to be linked up. The solution is, I think, it does involve national decisions about national standards and indeed national registers. And in cancer, we've actually done it with cancer registration. It's taken 20 years. Europe has managed to do it with the Register of Clinical Trials. That took 15 years. So I think this will happen, and I hope that it is in my lifetime, but I turned 61 yesterday, so we'd better get a move on.
Well, I think everybody will say happy birthday, Richard, albeit a little bit belatedly. So thank you for that. Realistically, we're probably not going to get through all of the questions that have been submitted today, because there's quite a few on the discussion panel. One of the things we can look to do is take those questions and always answer them afterwards if we don't get to it today. So the next is, who is ultimately accountable for the low sample use rate across the board? Does this sit with the biobanks or with some higher regulatory body that needs to be held responsible?
I think the challenge is that it's not known, and it's also not necessarily even perceived as a problem. So if you're operating as a research tissue bank, you'd apply to the HRA to get permission to collect those samples. You set out why you need them, you set out the consent, you set out all the various things you need to have them. But nobody actually checks if you're then doing that. And that's why we are trying to put the message out that non-use is a misuse, because it's almost perceived as, well, it can't be a misuse if they're not being used, because you can only misuse something if it's been used in the wrong way. And it's trying to get people to even recognize that this is something that needs to be addressed. It doesn't really sit with the HTA because it's not really within the Act. So it's unknown, I guess, is the question. I think the responsibility for trying to make sure samples are used does sit with the biobanks. But whether there's somebody accountable for that in terms of to be checking and seeing what's happening and asking the questions, that role is not defined.
I also think that from a certain extent, it's everybody's responsibility and everybody should be accountable. As Richard said right at the beginning, that, you know, it's a very privileged position where a patient's donated their samples. Sometimes it's not a particularly nice process to go through for that donation, and I think it's everybody should be taking responsibility to make sure that that is used as much as possible. The next one goes into a little bit more around the use of samples. So this is submitted from Robert. Thank you, Robert. Can the panel please comment on the current level of demand for samples coming from SMEs? Given that many UK biobanks have closed down due to the pandemic, how are the samples being sourced? Helen, not to throw you on the spot, do you want to have a stab at that?
Yeah, happy to. So there is still a massive demand. I mean, everything we've worked on for the last 2 years has shown us that, you know, that this is that SMEs do need these clinical samples, and that there's still requests coming in to me, you know, several a week from companies who are still wanting access to samples. And yes, admittedly at the moment there's could be issues around you know, the sort of logistics of onward transportation of those samples, particularly if they are coming from overseas organisations. But it's R&D isn't stopping just because we're in the middle of a global pandemic, albeit it's quite a challenge of its own at the moment. I mean, I would say that just overall, if we kind of park the COVID-19 for the moment, I mean, going forward, they are a valuable resource, and samples are already identified for the SME sector in order to progress their R&D assets, and they need to reach their programme milestones, they need to get to their investment decisions. But the other thing that's happening in the next couple of years, with the new in vitro diagnostic regulations coming in the middle of 2022, certain categories of device that weren't previously under regulation are now going to be in scope. So you could see this leading to an increase in the sample needs for things like validation purposes.
Yep, and certainly from our perspective, obviously we facilitate researchers placing those requests. We've not seen a drop in the number of requests that are being placed. We deal with small biotechs through to global pharmaceutical companies based in all different regions of the world. Obviously, there's an immediate focus on the COVID-19 samples, but we still have the standard number of requests coming through the platform on a regular basis. And I think that ultimately, as what we've seen the researchers do is they're looking, there's two things that we've noticed. First of all, they're still casting a wide net to see who has the samples, but then they're also being a little bit more cautious with regards to where the samples are coming from, i.e. where the location of the samples will be shipped from, just because of the problems with border controls and shipping and things like that. So that's something else that normally they don't focus on too much within the pharma side. They're looking for the highest quality samples with all of the appropriate due diligence and compliance requirements attached. But that's certainly something that we've noticed as well. To continue, to see if we can get through as many as possible, a question from Alison. Do we have too many biobanks? Should there be a small number of UK centralised facilities that act as a biobank service to supply samples? Possibly a bit more of a controversial idea. I will pause to see if anybody wants to take the first pass.
So I think this is a question that we do need to address, particularly around issues of sustainability. So I think there's clear evidence from the work that we've done that researchers really value a local connection. So when they're trying to find samples or trying to start up a new project, they really value the fact of having somebody local that's around the corner that they can talk to, discuss what they need, and really kind of establish that route. But then at some point, that project may stop, and then where do you put them and how do you sustain the system longer term? So I guess there's two aspects. One is the low utilization rate because there's too many already and there's not enough demand? Or do people just not know about them? And if we made them more visible, would the demand utilize them? And I think generally speaking, we do need to have more centralized facilities. I think we do need to think about the sustainability. It's clearly not sustainable within a single university, for example, if there's a dozen biobanks, all with their own freezer, alarm systems, staff, call-out systems, that can't be sustainable. So we do need to look at that. And I think that's one of the debates that we really need to have is how long should a biobank be operating? How long should it be collected, and when should it stop? And those first, the start-up of a collection is kind of the bit that gets people going, but it's a much harder conversation about when does a biobank stop. Thank you.
I know we're coming up to time, so I'm going to wrap up with the last question. Apologies, we haven't got them all of them. We will finish with this one though. We're getting a lot of different bodies asking us to give them info about the samples in our biobank. We want to know which one is going to be the one in main use.
Phil here. So what I would say would let us know of those examples where you've been asked for multiple. Our general view is that once you register in the directory, we can make that data then discoverable or port it into other systems as needed. We've done that, for example, with Health Data Research UK and their Innovation Gateway. We've also done it with Biobanking. So we're trying to reduce duplication for those types of systems. So let me know, get in touch, I'm sure my contact details are around, of where that duplication exists, and we can try and help reduce it, because if we're reducing it for you, we're probably reducing it for several other people as well.
Thank you. And actually, Phil, just the one of the last questions that just popped in, just for clarity purposes, for the TDCC, are you funded by the UK government?
So we're funded via the UKCRC. We're administered by the Medical Research Council, but our funders are, we've got, for example, so Medical Research Council would count as government, but we've also got the Wellcome Trust, we've also got Cancer Research UK, British Heart Foundation. So it's a mix of funding from across multiple funders that saw the need for a tissue directory. But our headline administrator of us is the Medical Research Council.
Sorry, this is Richard here. Sorry,
Hi, Richard.
I am going back to that. Some of those funders, like Cancer Research UK and the British Heart Foundation, get their money directly from the public. It's our money. The government gets its money from us. It's our taxes. We are talking about our samples that we have donated to benefit fellow patients and humanity in general. That's the issue. It's not happening fast enough.
Sorry, Richard, didn't mean to cut you off there. So that completes our questions for today. So we're going to go ahead and wrap up the webinar. Thank you so much to all of our presenters for taking part and for sharing their expertise and knowledge. It's been greatly appreciated. If you have any additional questions or you'd like to get in touch with us or any of the presenters, you can see the contact information available on the screen today. This webinar was also recorded and we'll make it available for later listening. So with that, thank you so much for joining the Scientist.com and the UKCRC TDCC webinar, and we look forward to seeing you at our next webinar in May this year. Have a lovely afternoon, and I hope everybody stays safe and well.